So we'll move forward with our next speaker is Dr. Timothy Foley. Dr. Foley, he is the DNA-encoded library selection and primary pharmacology lab head at Pfizer. He will be talking us today about building the efficient discovery of actionable chemical matter from DNA-encoded libraries. We're very excited to have him here today. We're looking forward for his talk. Tim, the stage is yours.
Thank you. Thank you very much for the warm introduction, Serene, and the invitation to speak. I really enjoy this venue, this organization, and the Assay Guidance Manual. It is what got me through graduate school and eventually my job here at Pfizer today. So as always, happy to be giving back to this community. And I hope you all are taking home as much from this short course as I am. So today I'm going to talk to you about our effort at Pfizer to develop the efficient discovery of hits from DNA-encoded libraries. And so I'll start with a brief introduction to what DNA-encoded libraries are. This is a relatively new technology developed within the last 20 years and has really caught on in the last decade. And so these are a collection of compounds that are produced through multi-step combinatorial synthesis, where each molecule in the library, here we commonly refer to them as the warhead, is appended to a DNA tag, covalently appended. This is a strong chemical bond that can't be broken, to a DNA tag whose sequence describes the building blocks that were used during the combinatorial synthesis, so that at the end of the day, you have a chemical structure and a DNA barcode whose sequence describes the chemical structure on the end. Now, these are produced through what is called a split and pool paradigm, so that the way that these libraries are built is that the small pieces of DNA of each individual DNA piece are arrayed in a multi-well plate. The chemical reaction is conducted on the warhead in each individual well with a different monomer in every well. And then the DNA tag is ligated to the DNA tag. The encoding is done to ligate the DNA tag on. So at the end of that round of chemistry, all of the wells of the plate are pooled together into a single tube, mixed up efficiently, and then redispensed into a new multi-well plate for the next cycle of chemistry. Now, what that means is that every possible combination of monomers that's in your library exists in, or is theoretically going to be produced into the library that you get at the end. So we call that, we refer to that as the full combinatorial cross. You can think of this like a Punnett square for the biologists in the room. You just keep adding dimensions and you get every possible combination. So we go to great lengths during the library design and through rehearsal reactions before the library is produced to maximize the congruence between the DNA tags and the expected products. And that's to say, we may design a library to have a couple thousand monomers in it, and then we subject them to a test reaction and ask, under the explicit conditions that the reaction is run, do they actually react? Now, that's because to do organic chemistry in the presence of DNA means you need some water around. And so the types of conditions that are used might not allow all possible monomers to be incorporated into the library. And so we cull the monomer list before the library is actually produced, so we eliminate those that are not productive. And so we go to great lengths to make sure that the code should have the molecule that it describes on the end of the library, or on the end of the DNA in the library. Now, this is a talk about assay guidance and screening, and so in this, I will cover how DNA-encoded libraries are used, and so not so much about how they're made. That's it as far as how they're synthesized. There are plenty of great resources out there for how DNA-encoded libraries are made if you are interested in learning more about them. So to say, at the end of the day, since everything is all pulled together in a single library, it's effectively in a single tube. And then we take a bunch of different DNA-encoded libraries that either have different monomer sets that were used for their production or different chemical reaction schemes that were used to put them together, and we mix them together into what we call a Dell library pool. And so the entire Dell library, entire collection is in a single tube. And here we see a picture of me holding Pfizer's first shipment of DNA-encoded libraries. There's enough Dell in that little white bit on the end of my thumb to conduct 5 full high-throughput screens of the Dell library in a single tube. So we identify hits using an affinity selection process, where first we combine the target that we're screening along with affinity beads in the Dell library into a well of a plate. After we allow that to come to binding equilibrium, these affinity beads are typically magnetic, and so we use a magnetic apparatus to remove the beads with the target protein and the bound Dells out, subject them to a couple of washes to remove non-specific binders, and then transfer them to another well on the plate and heat them to melt the protein and denature it and release the specific ligands that were bound. And so when we do these selection experiments in practice, this process describes 1 round of Dell selection. We typically conduct 2 or 3 rounds of iterative Dell selection, where we take the specific ligands that are released from 1 round and use them as input for a subsequent round of affinity selection. So we get a couple of iterative rounds of selection, and that's required to drive the background signals down low enough such that we can clearly discern the true hits that are in there, the compounds that are being enriched repeatedly through this process. So since 2017, Pfizer's been collaborating with HitGen to synthesize a Dell company in China to synthesize Pfizer's own Dells. These utilize Pfizer-defined chemistry and our monomers to give Dells that are in attractive chemical property space. So we're looking for molecules that make good starting points for our drug discovery projects. And so you can't just put everything in the tube. You might, we're interested in molecules that look like they would be something we would want to process into a drug candidate. And so we were designing our libraries purposefully in chemical space to possess drug-like properties. Now, what we like about Dell is that it has a small reagent requirement compared to traditional HTS. The libraries themselves are unbiased versus any previous organizational therapeutic areas of focus. And so, for instance, if your company or organization had worked very heavily in neuroscience, your chemical collection might be biased toward the physical chemical properties that are required to cross the blood-brain barrier. And so you wouldn't have a full representation of all of chemical space. So we like that the Dell libraries are synthesized in mass and don't have a bias based on what your company's been doing previously. There's very short assay development time. I'll describe some of that to you in this talk. But relative to an HTS, the assay development time is short, as is the screening cycle. It generally takes just a couple of weeks to go from a validated Dell reagent to go through the screening process, conduct the sequencing that's required, and do the data analysis. And finally, the last thing we like is that since this is miniaturized into a single tube, that we can conduct multiple screens in parallel to build a very biologically rich data set that can allow us to focus onto the compounds that we're interested in and looking for in the screen. So I'll start by describing what does it take to be a hit in Dell selection. And so going all the way back to first principles, KD rules everything around us. This is effectively a small molecule immunoprecipitation experiment. And so to think back to first principle, it's important to me to think back to first principles and ask what should we expect based on the biochemistry that's going on in that tube for binding, and what does that tell us about the types of hits we get out the other end? And I'll say that the affinity selection that is used here in DNA-encoded libraries, the principles also apply to affinity selection mass spectrometry, another emerging technology in drug discovery that is being used by a number of other pharmaceutical companies. So, but the concept of affinity selection is still the same. And in the end, very similarly to HTS, what we're looking for is to occupy the receptor with a ligand, whether that ligand is an inhibitor or an activator, that you have a receptor, your protein or drug target as a receptor, and the small molecule is the ligand. Now, there are two ways that you can do that. However that happens, one of the two parts of the binding reaction has to exceed the KD of the interaction for a high fraction of the receptor or ligand to be bound. And in HTS, we typically run the ligands or the proteins and receptors at a small concentration. And then we vary the concentration of the ligand and see at what point does that receptor become saturated with ligand. That would be in discerning a dose response curve. And so we could say that the fraction bound is related to the binding constant KD and its ratio the ratio of the ligand relative to that KD. Now, in affinity selection, it's flipped. This is because, and so in affinity selection experiments, the ligand is kept at a very low concentration, and then the receptor concentration is varied up and down the KD. So affinity selection is driven by receptor concentration, where HTS and traditional screening are driven by ligand concentration. So there's going to be there's going to be a relationship between the protein concentration that you use in the experiment and the affinity of the molecules that come out the other end as hits. We exploit that when we design our screens, and so we can run since we can parallelize multiple screens at the same time, we can run the Dell screen at multiple protein concentrations and get an idea of the relative affinity of the compounds that are in the hit list by whether or not they're present. And that can tell you about whether or not the receptor concentration that's used is above or below the KD for the compound. So for Dell selection, the compounds are typically held at a low constant concentration across all of your conditions. In practice, that's somewhere around 1 million copies per molecule. So not that many molecules. You put that into molar, and it's somewhere below attomolar. So that's 10 to the minus 15. So they are not participating at all in the determination of how much fraction is bound in the experiment. So, and here I'll describe Dell selection typically being tested at 2 concentrations that allow you to bucket your hits based on affinity. Now, when you do that affinity selection process, the enrichment that you occur, which is how much you've decreased the background, typically occurs on a low range about 200-fold per round of selection, and on the high range, somewhere around 500-fold. And so if you take a Dell library with a few 100 million compounds in it and put it at 1 million copies per compound in a tube, you're going to need to do a few rounds of selection in order to get the copy count down to what you can load in a DNA sequencer, which is somewhere around 100 million to 1 billion total DNA copies. So we find that if by either metric at the lower recovery rate, we typically conduct 2 to 3, sometimes 4 rounds of selection in total to get the background down to the number of compounds that we can put on a DNA sequencer. And what we see in practice based on this, the relationship between this affinity, the conditions that we use, and the first principles of biochemistry very much confirms what we expect from the first principles. And here we see a simulation of a population of molecules with varying affinities for the receptor in the experiment, and then looking at a simulated Dell selection experiment and seeing what portion of the population is composed by the various affinity molecules. And so you can see for the non-binders, which would be compounds with a KD around a millimolar or even 100 micromolar, that they are very quickly washed out over the first couple rounds of selection, at which point the molecules that are in your tube become dominated by compounds with affinities below 10 micromolar, and that you could eventually run enough iterative selections if you wanted to wash out all of these and only arrive individually at the compounds with the highest affinity. But in practice, what you do is decrease the total number of DNA molecules in the tube such that you can't even run a PCR to amplify the sequences that are left. So there's some practicality involved in deciding how many cycles to do. But what we see is somewhere between 2 and 4 rounds of affinity selection or compounds that come out under these conditions that our hits are typically in the single-digit micromolar range or better, that we don't see much that confirms above that. So obviously you could increase the protein concentration if you wanted to discover hits with weaker affinities, but in practice, this is how we're doing this. So after we had begun working on Dell, after a couple of years, built our own libraries and begun testing them, we got some feedback from our project teams, and this is what I want to talk to you about today, is how do we do Dell discovery efficiently? And the feedback that we got was that sometimes we run a Dell screen and we don't find anything at all. And that led to questions about, is Dell selection itself a reproducible process? You can write an equation on the page based on first principles, but does the operation of the experiment actually reproduce that? And is this a robust technology, or is it much more finicky? Like, would we get a different result if we repeated the screen? And so I'll talk to you a little bit about that. And then when we do find hits, they have poor properties. So our medicinal chemistry team is not terribly interested in optimizing compounds. But having a program start with a compound in really bad property space, it's much harder to make it better than it is to make it worse. So in effect, the hits that we were finding with Dell were disadvantaged relative to compounds that were arising from other discovery technologies. And three, that the hits, when we do find hits in the signals and the data, when we make them off DNA, they don't work. And so the talk will focus on these three main points from here on out. So we initially started and wanted to address the question, well, how reproducible is a Dell screen? Since we were running some and they weren't working, others were. And so I'll talk to you now about a series of experiments that investigated the reproducibility. And so here we see the results for a Dell screen. This was from a library that had 3 cycles of chemistry. And so we visualize this data in a cubic plot. This is a plot in 3 dimensions where the identity of the monomer for each cycle of chemistry is assigned to a different axis. And so that each compound within the library has a unique physical address in 3-dimensional space. And so that way we can plot the data in this 3-dimensional space and scale the size of the data. All right, I'll turn my laser pointer back on. I must have been a little overzealous with my elbow and my phone, my apologies. So the cubic plot. So without an understanding in the literature about how reproducible is a Dell screen, we looked to repeat the experiment and see, could we discover this green line? Since this green line, we had confirmed that the chemical structure described by the DNA barcodes that make up that line, in fact, is a true ligand to the protein. And so with that, as part of our technology transfer coming from HitGen to bring not just the DNA-encoded libraries, but also the capabilities to screen with them into our internal Pfizer laboratory, I traveled to China and repeated this Dell selection campaign under their supervision to demonstrate that my technique was sound and that I was taking back a confirmed and valid process to Pfizer in the United States. And so what we see here is the cubic plot from that repeat experiment. And we were elated to see that both we had reproduced the discovery of this compound series, because as you see in the green line here, that we were able to capture it nicely in this experiment, but also the relative size between the data points within the lines appears to be reproduced. Now, that is probably best looked at as a scatter plot of the first screen against the second, shown here in the lower left panel, where the data are colorized and then in an analogous manner with the hit feature, data points arising from the hit feature being colored green. And so we see here between screen 1 and screen 2 is excellent linear correlation between the hit feature and a much less convincing correlation between the blue data points that make up the rest of the compounds in the background. That are the background signal of this screen. And so with this knowledge, I returned to the United States and set up our capabilities in our labs in Groton and repeated this screen a 3rd time. And so what we see here is the cubic plot from our 3rd screen that was done in our Pfizer labs. And again, we were able to rediscover the same line feature. and that when we compared the correlation between screen 3 and screen 1 or screen 2, that both of the hit feature data points correlate very nicely. And with the exception that in the repeat that was done at Pfizer, you'll notice the orange line in all of these graphs represents unity, that the data cloud in screen 3 is above the unity line. Now that was to be expected in the experiment because of the way we did the DNA sequencing. So we were able to sequence a lot more of the sample in our laboratory at Pfizer, and what that did was allocate more screen reads to the sample, and so we would expect hits that are present in there to be observed more times than they were in the other two experiments. That's exactly what we see here. So with this combination of results, we were quite confident that the underlying binding assay that underlies Dell selection is a reproducible and robust process. And so with this analysis and capability in hand, we then look to address the question, where are the pretty hits? We built our libraries around drug-like properties, and why aren't we finding them? And that had to do, the way we approached answering that question was to apply the molecular property optimization scoring, which is a subjective strategy to assess the relative balance of drug-like properties between individual compounds within a data set, often used in the optimization of compounds for drug-like properties. Then there are medicinal chemistry optimization from a hit toward a clinical candidate. And so the way that this works is we assign an individual score value between 0 and 1 for a collection of different molecular properties. Here I've got 3 listed that were important to us. One, the molecular weight. Compounds get too big, then they don't obey the ability to passively diffuse across membranes. And so starting from low molecular weights get a high value of this score, and then it's graded down to a value of 0 at high molecular weights. Similarly, for the log D, which is a very similar parameter to the partition coefficient log D or log P, and then also the count of hydrogen bond donors within the molecule. And so we can take the individual scores for each property and sum them together into an MPO score, and look at our data based on the value of this MPO score. Look for the compounds with the highest numbers. And so the way that we do that in this cubic plot, I'll introduce you to this line feature here, which was produced in a Dell screen of BRD4. That's a BET protein that binds, then the bromodomain of this protein binds acetylated lysines, a very well-known, well-characterized ligandable target. So we use this as a model system for a lot of our Dell experiments. And so here we see a line feature that was produced from screening BRD4 against our Pfizer Dell libraries. And this is plotted as I had previously described, where the data point size is proportional to its the number of counts that we saw in the DNA sequencer, which is the signal in the assay. And then the data points are colorized by this MPO score in a classic stoplight pattern, from low values being red to progressively values of increasingly positive values, eventually passing through yellow and into green. And so what we see by looking at the entire feature that's there is that the largest data points in the screen are the compounds with poorer molecular properties. Now, while it was seemed fairly intuitive, at least initially, to go after the compounds with the biggest signal because they're going to be your best chance of confirming a hit, in fact, those aren't the molecules we wanted. The ones we wanted were the ones that still made signal but had good properties. And so I show it to you here using properties as this MPO score as an ability to filter data based on the properties. I'll say that in practice, we use this more as a tool to guide our selection of a compound, not as much for filtering, but to look for who has the best signal and the best balance of properties. And so, yeah, not used as a filtering tool, but it makes for a really great image here. And so what you see is if you take this line feature and apply a filter to that MPO score to only show you the green data points, we can now focus in on the large data points that have the best molecular properties and pick those compounds for follow-up. And so we now have an excellent way to get to the compounds that are the starting points that our medicinal chemistry teams want out of the Dell screen. We still had a challenge that even with this tool, the hits, sometimes when we make them off DNA straight out of the screen, they don't confirm. And we wanted to ask a couple of questions why. So I'll talk to you briefly about assay development here and reagent validation for Dell screening. And say in the context of assay development, the established technique is typically to take your protein, run an assay in solution, then immobilize your protein to these affinity beads and run an activity assay of some sort. In this case, for BRD4 that I'll be describing, it was looking at the polarization of a fluorescent ligand. And so it's a simple binding assay, runs an HTS really smoothly. And so what we see is the signal window here for this BRD4 assay in millipolarization units for the protein in solution versus when it's immobilized to the bead. So we see that the signal window has been greatly compromised. Now, when we check the dose response, the pharmacology of that target when it's in solution versus immobilized, that appears to reproduce. Both of these curves display the same IC50 value for the compound, so that told us the pharmacology is preserved for this target. But what's up with that signal window? Is that because of some interference of the immobilization beads on the readout of fluorescence polarization? Or is it because 75% of the targets just got denatured as they bound to the beads and they in effect don't bind the tracer at all? So what sense do you make out of this as a looking for a robust assay and technique to validate our targets? This is not the best way to go. It's not a quantitative technique. And so what we developed was a workflow that we refer to as bead-assisted ligand isolation mass spec. This is a technique very analogous to the Dell affinity selection process. And so this takes a receptor and magnetic bead, you bind the receptor to the beads, you add the ligands, and then perform a simple magnetic separation and ask, where did the ligand go? Did it stay in the supernatant, in which case it was not bound by the receptor, or did it partition onto the beads? And then to put both of those through a detector. Now, we took the same fluorescent ligand that was used in the fluorescence polarization experiment and conducted a titration of the ligand against a constant concentration of 500 nanomolar BRD4. Now, under these conditions, with the affinity of the warhead on the end of that tracer, we would expect binding to be stoichiometric. This is the protein concentration far exceeds the KD, so every molecule of ligand that you put in that solution is going to hit the receptor and stick. And so what we did was effectively turn this into an active site titration experiment. And so what we see here is the fluorescence value of the eluate from the beads and the fluorescence value from the supernatant of what was left. And so what we see in both of these samples is an inflection point in the curve. And what we would expect is that if the compound is being captured, it would show up in the eluate, and if it's being left in solution, it's in the supernatant. And so we see very clean, complete capture of this tracer by the receptor, and it accumulates only in the eluate up until about 500 nanomolar. And after which we see direct linear accumulation in the supernatant of the experiment here, and no further capture on the beads. So this this inflection point between both curves indicates the total concentration of receptors in the solution or in the sample. And in effect, both of them agree that there's 500 nanomolar of receptor in this experiment. And that was directly in line with what we had observed through other classical active site titration experiments with this BRD4. Now, this was an interesting observation, and we said, well, this is great, but it's not generally applicable to all drug targets because it needs a chemical biology tool in order to do this. And so we said, well, if it works with fluorescence, it should work by mass spec as well, using classical techniques that are part of the ADME sciences. We should be able to quantify the amount of an unlabeled molecule that's left in these fractions using LCMS. We see here in this lower trace And these lower traces is looking at the LCMS signal that's produced by the bead fraction and the supernatant fraction from an analogous experiment with an unlabeled tracer. So see, again, nice linear accumulation of the tracer of the molecule, unlabeled tracer, in the eluate fraction of the experiment until it reaches the total concentration of active sites in the experiment, and after which further increasing the compound concentration doesn't lead to more binding to the beads. And similarly, there's no accumulation of the tracer in the supernatant of the experiment until we hit this breakpoint, and then there's linear accumulation afterwards. And this inflection point is the active site concentration in the samples. Now, between the two experiments, understanding that we wanted a slightly higher signal in the mass spec, we increased the receptor concentration. And so that told us that we should expect somewhere around 700 nanomolar of receptors in this sample. And the mass spec provides an additional level of detail in that as it's set up, it's calibrated to an external standard curve. And so that you can receive your data out of that instrument as either area under the curve or total counts by the mass spec, or transformed into units of concentration of the analyte. And so looking at where this inflection point lies for the unlabeled tracer in the signal of the mass spec that's been converted into concentration, again, is at 700 nanomolar. So between all three of these different ways of analyzing this single experiment, all three of them point to a single concentration of receptor in the experiment. And so we have made the simplicity of this technique using the mass spec as a detector and its ability to report on the ability of the receptor to capture known ligands in an affinity selection process. This has become the cornerstone of our Dell reagent validation process at Pfizer. There's very little, if no, assay development time that's involved here, which means we can move through the process fast, because one of the things we like about Dell is that the screening process is quick. And so this is the cornerstone of how we validate reagents. And with these observations and confirmation of these types of behaviors in our samples, we feel very confident that if we take that into a screen, that we're testing the hypothesis of, do our libraries contain ligands to these proteins? And so if we do observe a result where we don't find any hits in the library, we can comfortably accept that this target is either undruggable or the chemical space that overlaps that define its binding sites does not well overlap our Dell libraries. And then in the last few minutes here, I will talk to you about really how we fix this problem. So now we can get into an experiment and say, do we believe we tested the hypothesis when we don't find hits? And then how do we make sure they confirm? And this had to do really with a paradigm shift in our philosophy around Dell, which was that Dell barcode, even though we worked really hard to make sure through library synthesis, the warhead on the end represents, at least in some major portion, the molecule that's described by the barcode, that's not always the case. And so here we see an example Dell synthesis scheme for a 3-cycle library, where this is Marcouche structure of what we would expect out on the end of the barcode and the molecule we would be focusing on if it appeared as a hit. The reality of the situation is that either through failed chemistry or side products, any of the molecules drawn on this slide could be formed in the chemical reaction that is the chemical sequence that defines this Dell library. So no matter what's out on the end, all of them have the same barcode. So not one DNA barcode represents one small molecule due to the possibility of failed chemistry, reactivity relationships, that all of this needs to be taken into account over the course of the Dell screen. And this is really, again, to focus on the low confirmation rates of our Dell screens, where this was our prior workflow, where we would run the Dell screen, see a feature, make a molecule, tested biochemically to see if it binds, and we'd be crossing off series left and right. In effect, very, very few series were confirming or requiring a very high resource commitment from our chemistry teams in order to find a few hits, and that we weren't able to interrogate everything in the library, all the hit signals. So responding to this feedback was that we had this growing confidence in the technology, And that with that, we understood something in the Dell has to be making this signal, and could it possibly be these side products? And how do we efficiently triage that? And so about this time, there was a talk at SLAS 2018 from Joe Franklin at GSK, talked about their high throughput binding conformation platform, where they apply on DNA resynthesis with cleavable linkers to characterize the chemistry that actually happens with those combinations of monomers under the chemistry synthesis conditions, and then defined their the true ligand that's responsible for the signal using by cleaving the molecules off the DNA and conducting an ASMS experiment. Now we had built our own on DNA conformation platform, and so I'll focus again here to describe that to you with this BRD4 example. So here we see a compound that produced signal in that BRD4 screen with reasonable chemical properties. It doesn't make anybody cringe when they look at it. And Anoka went back and resynthesized this, and while attached to DNA following a library procedure. And what we see is the HPLC trace of that on DNA mixture. And surprising to us, what we saw was none of this title compound attached to the barcode. And in actuality, two different compounds that were both side products of the reaction conditions were present in reasonable yield. And so we see this compound 1 that doesn't have the first blue building block, and compound 3 that represents the fully elaborated library molecule. But both of them have a second incorporation of the third building block that occurred because of the way the library synthesis was designed, that this functional group is reactive in an analogous manner to the aromatic chloride that would be used here for the nucleophilic aromatic substitution reaction that's used to incorporate this building block. So what you do is you get two of those building blocks attached to the molecule. So this is great. We can say, well, this feature doesn't look like it represents the chemical structures we were expecting. We took it a step further, and through analyzing the chemical reactions, we understood that the LCMS we were using to analyze these molecules attached to DNA had the sufficient sensitivity to report on a biology experiment. And so that we could effectively take our BALI-MS platform that I had described to you before and repeat it with these on DNA compound mixtures and fish out the true binder out of that without having to install a chemical link, a cleavable linker to chop the small molecules off the end of the DNA beforehand. And so what we saw when we took this resynthesized compound mixture that I described on the previous slide and selected it with a mobilized BRD4 and looked at what was in the eluate and compare it to a beads-only sample, we see that the both compound 2 and compound 3 are enriched considerably relative to a beads-only control. So what this tells us is that both of these compounds are, they both bind the target, and the additional Dell signal that we observed, that data point, was the sum of contributions of affinity selection of both of these compounds from the library. And so in effect, this has been the revolution in technology that we needed in order to become efficient with the technology, with the Dell technology. And so what we've done is, instead of remaking molecules directly through off-DNA synthesis, since we understand it might not actually be the molecule on the page, we've incorporated this on-DNA conformation, which includes resynthesis, chemical characterization, and binding studies, which takes us about 4 or 5 weeks to reproduce, to point us at the molecules that have good properties and point us at the molecules that are actually making the signal in the experiment. And what that has done is greatly increased the number of series that, when we make them off DNA, that confirm. And that makes our project team happy because we're not wasting their medicinal chemistry resource making molecules that don't confirm. And so with that, I'll summarize here in the last minute of my allotted time that, you know, I hope that what you take home from this is combining robust reagent validation and this on-DNA platform, that combining that together makes an efficient workflow for delivering actionable chemical matter to project teams, and that the binding assay that underwrites Dell selection, that it obeys first principles and is a robust and reproducible experiment. So I will pause on an acknowledgement slide to say thank you to the HitGen and Pfizer, the HitGen team and the Pfizer Dell team for all their work here in helping us build what is what I feel to be a world-class discovery platform that uses DNA-encoded libraries. So with that, I'm happy to take any questions.
Thank you very much, Tim. That was an awesome and informative presentation. So you do have multiple questions, and I think we can erode into the breakdown to answer them. So let's start with the first one. So this question is from TC, Dr. Thomas Chang. He's asking, is the final release of specific ligands also done with pH, chemical precipitation? And he follows that with, what is the Goldilocks rule for rounds of selection? And TC, if you would like to add anything, please feel free to unmute yourself.
Trying to find my way into there. So the first question was revolved around the screening itself or the library synthesis?
No, just the release. Is the release always done by heat, or do you also have chemical precipitation or pH change and shift?
Oh, to release the compounds from the beads?
Yeah.
Yeah, so what we find, and you'll see this described in another paper I published at the beginning of 2020 with Justin Hall and David Israel, was a peculiar observation that when you do the Dell selection, you get a decrease in the number of, if you take the beads and bind the library and you keep washing them, eventually you hit a lower asymptote where you can't wash anything more off. And then you, in order to get and then we heat release. I think there's something more going on there that has to do with the DNA tag interacting with the bead. So you get the molecule targeting the Dell molecule, the receptor targeting the Dell molecule to the bead, and then you get the DNA tag forming some secondary interaction with the bead. So it doesn't like to come off the bead until you heat it. I haven't looked. There's details in the literature for other papers, people using different chemical and biochemical treatments to release the molecules in solution. But what we find by and large is the heat process is reproducible and effective.
So that's very useful, Tim, because yeah, the high negative content of DNA is almost like a screen. So that's great. And my second question, just in terms of your experience, is it always 3 rounds of selection, or do you find that there's actually a Goldilocks rule? You know, some libraries can do fewer, or some you have to just get it right, right?
Yeah, so I think that, and you titer it by, and that's really the job of the selection biologist, is to, between each round of selection, you titer what eluted by qPCR, and you say, how much do I have left? And after the first round, you can guesstimate what the relative enrichment is round to round. And you can say, if I do 3 washes, I can expect this to go down 500-fold. And when your sweet spot is really about 3 times 10 to the 8th compounds at the end of the screen to do all the molecular biology for sequencing. And so that's what we focus on getting. And it has an error around it of about what's really usable is somewhere between 3 times 10 to the 9th and 3 times 10 to the 7th. So you get a pretty wide target space to hit, but you can watch this and say, if I'm at 3 rounds of selection, if I do a 4th with the number of washes that I did previously, will I get there? Or will I overshoot? And then for the last round, you could back off a wash or 2 and to decrease the enrichment and hit it within the range that's required for molecular biology. In practice, what I think it has to do a lot with your target. How sticky is your target, and possibly even what is the relative surface, are there patches of positive charge on the surface of your protein? Is it non-specifically enriching DNA molecules? Now, your buffer does have a whole bunch of excipient DNA, salmon sperm DNA, that's added to prevent non-specific interactions. But in practice, we see that just some targets non-specifically enrich the Dell library more than others. And so,
actually,
we're running 2.
Yeah, so actually what you're really saying is that you can do the experiment, you can actually do a round of selection, see what you get, and go to second, third, fourth, and you can actually test at each point. Thanks.
Yeah, well, it has to do with also population sampling statistics. I'm happy to take this offline or wax philosophical for a really long time. But you know what and it's described actually in this paper that was accepted at S/Discovery last night. So coming very soon to an inbox near you will be this paper from me. And effectively what you need to do is sample enough of the hits in order to see them above the background. And if you stop early and take a very small sample, it's sort of like, as my statistics colleague likes to refer to it, is trying to find the Satanists in the room. And so you've got to ask, you know, if they're at a very low concentration in the population, you've got to ask a lot of people before you find one. And so you need to sample a pretty good portion of the population in order to get it.
Thanks, Ken. Didn't mean to dominate.
Okay, thank you very much. So we have only like 4 minutes, so we'll let people take that break. Tim, you do have 2 more questions, so if you don't mind going to the chat box and the Q&A and answering those questions directly, that would be awesome. Again, thank you so much for an awesome talk, and let's take a few minutes break, and then actually it's at 3:15, so it's my bad. We do have maybe we'll take one more question, and then let's take this question. So this is from Yaming Wang. Thanks for the talk, Tim. Can you comment on whether the Dell technology has special advantages in application in certain target classes over others?
Do I think there are cases where Dell works better than classical screening? I think non-classical targets, it all depends on the overlap of chemical space and your, of what's in your chemical libraries you're going to screen and what the binding site on your target looks like. You know, I'll take this opportunity with as many people are here to say, this is not a cure-all technology. If your target doesn't have a binding site, I can't will one into existence for you. And it really depends on the overlap of the chemistry that binds to your target and the compounds that are in your collection. And if they overlap, you'll find a hit. If they don't, you won't. And so I think we hope that the relatively large amount of chemical space that you can cover in Dell efficiently allows us the opportunity to find hits for targets that might not work by traditional HTS.
Okay.
I think it also has a good application to targets where you don't have an assay, or orphan targets that you don't know what they do, or don't have a ligand, don't have a substrate. It makes it very hard to set up an assay for those.
Awesome, Tim. Thank you. I'll drop in one more question here for you, and this will be the last one. So the question is, what is the advantage of Dell versus phage display library?
Okay, so phage display library, so while they're conducted in a very similar manner, save two characteristics. Phage display only does peptides, so you can identify and enrich peptide sequences, not small molecules. So that's one aspect, that one differentiator between the technology. So the peptides might not be the types of molecules you're looking for. And then 2, it has to do with the fact that there's amplification at each round of phage display. So you run your experiment, you drive it down to a small number, you propagate those phages into a new population and select against it again. With Dell libraries, there's no propagation step. And so you've got to start out at a higher level of molecules to get something out at the other end. So I think that the phage display does a more efficient job of applying selective pressure, because you get that amplification and in the repeated rounds of phage propagation that you don't get with Dell.
Okay, thank you so much, Tim, for taking all these questions. We will take 5 minutes break now, and we will reconvene at 3:15. Please join us back. We have really exciting 2 talks coming up afterwards. One is on antibody therapeutics, and the other one is on COVID-19 and therapeutics. So we will see you back in 5 minutes. Thank you very much.